Hey guys, we are Group 11. Today we'll be talking about how we tested if phosphorylation of MDH affects its interactions with CS in Trypanosoma brucei.
So first, we're going to talk about our model organism and why we're looking at this organism. So we're looking at the parasite T. brucei, which causes human trypanosomiasis or African sleeping sickness. And this is a disease that kills over 10,000 people a year. And T. brucei are transmitted by infected tsetse flies to humans. T. brucei is present in different host organs at each stage in its life cycle and thus has different nutrient availability and needs at different stages. And as a result, they have different metabolisms and thus have enzymes that serve different functions to meet their current needs.
Metabolic reactions in the cell are often compartmentalized to localize enzymes to cellular regions with high concentrations of their substrates in order to allow for optimal use of the enzymes. Sometimes there are different isoforms of the enzyme in the different compartments in order to perform their distinct compartmentalized roles. This is something that we see with the enzyme malate dehydrogenase in T. brucei. During the different life cycle stages, T. brucei express different forms of MDH and they perform differently, giving different potential avenues for targeting these enzymes' functions with drugs. It has been proposed that MDH forms a metabolon with the next enzyme in the citric acid cycle, citrate synthase. This is done so that the intermediate metabolite oxaloacetate can be passed from MDH to citrate synthase without having to float around until it comes in contact with citrate synthase. The negative oxaloacetate molecule is channeled through the positively charged interface into the active site of citrate synthase.
It has been proposed that in order to form this metabolon, there is an important phosphorylation site, shown here, that is present in some forms of MDH, but not others. This is proposed to enhance the interactions due to the negative charge of the phosphoryl group interacting with the positive interface of citrate synthase. In T. brucei, there are three forms of MDH, the sequences of which are shown here. There's the cytoplasmic, mitochondrial, and glycosomal forms of MDH. The mitochondrial and glycosomal forms of MDH in T. brucei have the same phosphorylatable residues, serine and threonine, in the same spot shown in this light green box, while the cytoplasmic form does not. Thus, we decided to focus our project on the effects phosphorylation has on the binding efficiency between these forms of MDH and citrate synthase.
Due to difficulties associated with using phosphorylated proteins, we will instead be using a technique called phosphomimetic substitution. This is essentially when an amino acid is replaced with another amino acid with a more negative charge. This emulates a phosphorylated protein. This substitution must occur with an amino acid that is phosphorylatable already. The first step in our project was to mini-prep plasmids containing the normal version of GMDH and MMDH, preparing them for mutation. This image shows a 0.8% agarose gel electrophoresis. The bands in lanes 4 and 5 represent the plasmid DNA. All three bands are close to the position of the 6 kilobase band of the latter, which means that they were cut correctly by the restriction enzymes. To create a phosphomimetic mutagenesis, we used NAB base changer to design forward and reverse primers that we would use for mutagenic PCR. We designed these primers to induce mutations that would cause the serine and threonine on both mitochondrial MDH and glycosomal MDH into aspartic acids, making them phosphomimetic.
We then carried out the mutagenic PCR and transformed cells using the PCR product. We then mini-prepped the plasmid to sequence, but first we had to confirm the presence of the plasmid DNA in the samples, which we did on this 0.75% agarose gel. The bands, as you can see in lanes 2 through 7, are all at the same position as the 6 kilobase band is on the latter, as expected. Now here are the sequencing results for our GMDH samples. The sequences contain the correct phosphomimetic mutations in the first two samples. The third sample didn't have a clean sequencing reading, and thus the mutations weren't complete. This next slide shows the sequencing results for our MMDH samples. Sample number 1 didn't have enough volume for sequencing and returned no results. However, both of the remaining samples that were sequenced had the correct phosphomimetic mutations.
We plan to purify the mutated glycosomal and mitochondrial MDH as well as purifying citrate synthase. After this, activity assays can be used to determine the purity of the mutated proteins. This will also help us to make sure that our mutations did not affect the activity of the enzymes. After this, we plan to perform a thermal melt assay on the various mutated forms of MDH with their citrate synthase counterparts. A thermal melt assay is an assay where a dye is mixed with a protein sample and then heated. This dye fluoresces upon contact with denatured proteins. As the sample heats, the protein denatures and the dye fluoresces upon contact with said denatured protein. This will give a readout for the melting temperature. If proteins interact, they will stick together, increasing the melting temperature. This will be used to see if our proteins form a metabolon and if the phosphomimetic protein shows a stronger attraction, as their melting temperatures will be higher. This data could be used to determine a potential drug target to treat trypanosomiasis.
That's the end of our presentation. Here are our references. Thank you all for listening, and we will now take any questions you guys have.